endophytes. And so we thought something's really going on with these bacterial endophytes in plants. They have to be important. So what we were doing, I was in the lab with Kate, my lab manager, a lab researcher, Kate Kingsley, Catherine Kingsley, and we were at a microscope. We were tracking bacteria in plants, and we figured out that they are actually going into the plant cells, going into cells, and we could see them going into the cells at the root tips. And we didn't know, well, they're going in, but how are they? Are they? What's happening to them? So we were trying to track them with a microscope using some stains that we actually developed ourselves, new stains using reactive oxygen that detect reactive oxygen, actually hydrogen peroxide. And it turns out when these microbes, these bacteria inside the plant, they cause the plant to put reactive oxygen on them as a way to control them and extract nutrients from them. So we could put these stains there, and then we could see the bacteria. And without those stains, we couldn't see them at all, but we were able to see them inside the cells. And we also were able to see how they go into root hairs. And we observed them going out of root hairs. And so when we put together the picture of what's happening, that the microbes are entering at root tips or just behind the root tips, just behind, in cells just behind the root tips, and then they were going out of the root hairs, we knew that something significant is going on. They're cycling through plants. And as it turned out, a group in Australia working just a little bit ahead of us defined the process of bacterial entry into root cells. They called it rhizophagy. Rhizo for root, phagy is a term that implies eating. So they were suggesting that these, and this is a group out of Queensland, Australia, that they were suggesting that these microbes were being essentially consumed by plants as a nutrient source. So we don't have credit for that concept. We really have credit only for studying the process over a number of years and showing how the process works, showing what happens to the microbes when they're absorbed into plant roots. Plant roots actually will secrete these sugars, exudates. They're nutrients that plant roots will secrete out of the root tips, and that will attract bacteria and other microbes. Some other kinds of microbes will be attracted to that sugars, those exudates, and they'll accumulate at those root tips. And then the plant will take them into the root cells at that point. And then when they take them in, they will use reactive oxygen to put on these microbes. And that will essentially oxidize the cell walls off of the bacteria or yeasts, or in some cases algae. And it will take the cell walls off, and then these bacteria become protoplasts. These microbes become without, in other words, they're cells, microbe cells without cell walls. And then they process those protoplasts inside the root cells, and they actually will trigger root hair formation. So you don't have any root hairs at all unless these bacteria, mostly bacteria, are